Hi everybody, welcome back to another video. In today's video, we will pick up where we left off from the graded potential and we'll start talking about an action potential. Okay, so how does the graded potential become an action potential? We said it has to hit threshold. We'll talk about all of that. Okay, but if you liked the video, if you learned something from it, consider liking, consider subscribing, and let's get right into it. So what is an action potential? Okay, what are some of the characteristics of an action potential? So an action potential is very brief, okay? It is rapid, it is long distance as opposed to a graded potential, which is very short distance, okay? And it is extremely consistent, okay? And it has a very, very large amplitude. The consistency here, by the way, refers to the magnitude. The magnitude is very, very consistent. That means it doesn't change every single time you launch an action potential, unlike a graded potential, which does change. Remember, based on the strength of the stimulus, it does change. And it also has a very large amplitude of about 100 millivolts, okay? When an action potential occurs, we are completely reversing the membrane potential. Okay, we are completely reversing the membrane potential, so we'll refer to what this means later. We will also note that the action potential, as it travels, it kind of propagates, okay, along the membrane, okay, but it's more so specifically along the axon. Okay, and also it should be noted that it is not decremental. Okay, it actually regenerates itself. It regenerates itself. So we'll talk about again what that means. Okay, but it's really important to understand that a graded potential is decremental. Okay, it is not long distance. It is not consistent, and it does not have a very large amplitude. Okay, and in fact, it does not reverse the membrane potential either. However, it does propagate along the membrane, and it is very decremental, like we said. It does not regenerate itself either. Okay, so these are some of the properties. Now, like I said, where does it occur? It occurs right over here, but in fact, it starts right here at this portion that narrows out. We'll talk about it. This one's called the axon hillock. Okay, it starts here, but for now, I just want to kind of take this entire thing and zoom in on it and say, what is it that is actually allowing this action potential to travel along this kind of membrane like this, all right? And what is it that it's allowing that is allowing it to regenerate itself? Well, if we zoom in on that kind of membrane, right, we'll come across two very, very important specific channels. And when you see these channels, I want you to always associate them with the action potential. The first one is called a sodium voltage-gated channel. Voltage, gated channel. So anytime you see voltage-gated channel, immediately associated with a action potential. Anytime you see a ligand-gated channel, immediately associated with a graded potential. That's from our previous video. So potassium, voltage-gated channel, is the other one. Gated channel. Okay. Great. So the potassium and the sodium voltage-gated channel, they have distinct shapes, all right? So if I imagine that this right here, this line here, is my membrane, right? And then right here is the extracellular fluid, and this is the intracellular fluid, okay? You will notice that these channels, in their name, they have gates, right? Those gates are voltage dependent. That means that these are the gates. And these gates, if you look at them, they're facing the inside of the cell, not the outside, but the inside, okay? They're facing the inside of the cell. Great. Now, these gates have names, okay? So for sodium, it actually has two gates, if you notice. It has this one over here called the activation gate, and it has this other one that kind of looks like a ball and chain, and this one's called the inactivation gate, or IAG. This one also has an activation gate, and you know what? Let's draw it a little bit more closed, like this, okay? Activation gate, but you can see that the potassium voltage gated channel has no inactivation gate. Okay, no inactivation gate. Now we'll come back to these in a second. Okay, so we'll come back to these in a second because we're going to talk about some of their properties and what they kind of allow for. But first things first. Remember how we said that all action potentials are very consistent in their magnitude, as in like they don't vary, right? Each time they reach a certain amount, and then that's it. Well, if all action potentials have the same kind of amplitude, right? How can we tell the strength of the stimulus? Because remember, when we were talking about graded potentials, right? We said that this was our kind of graph, and we said that the more plus signs, right, the greater the stimulus, then the greater the actual graded potential would appear on our graph. But for an action potential, how can we kind of tell the strength of the stimulus? Well, it's actually quite easy. You can tell the strength of your stimulus by counting the number of action potentials. Okay? So for example, imagine that this, okay, each one of these is an action potential graph, right? So we'll talk about what an action potential graph looks like. This is it right here, by the way. But if I were to kind of zoom out of this view, okay, I literally just keep zooming out, zooming out, zooming out, zooming out, zooming out. Eventually, I would just see a line. This line is a very, very zoomed out version of this graph, okay? And that line is one action potential, and it's the same as this thing, okay? Which means that in this scenario, I am seeing 1, 2, 3, 4 action potentials. In this scenario, I'm seeing 1, 2, 3, 4, 5, 6 action potentials. Okay, let's say I will have this scenario be a light touch and this one be a hard punch. Okay. I'm going to ask you this question again. If all action potentials have the same amplitude, right, the same kind of magnitude, then how can we tell the strength of the stimulus that was applied to us? Well, I have one scenario here where a light touch to, for example, my arm. I touch my arm very lightly, and it produces 1, 2, 3, 4 action potentials on a graph. And then I really punch my hand extremely hard, and it gives 1, 2, 3, 4, 5, 6. Which one was the harder stimulus? Or the more stronger stimulus? Well, I think the answer is pretty clear. The one that produced the more number of action potentials, okay? So to answer this question, we can say that the frequency, or the rate, right, or the number of the action potentials tells us about the strength of a stimulus, okay? If the stimulus was extremely strong, I would produce multiple action potentials. If the stimulus was not so strong, then I would produce less action potentials, okay? So that's how you can tell. Now, another thing to just really kind of recap what we were talking about with the graded potentials is, let's say I have a random cell, right, a neuron. Let's say I have this kind of neuron right here, right, and I'm doing some kind of experiment on it, okay? I'm recording that experiment on my graph. This graph is membrane potential over time, right? This graph is the strength of my stimulus over time, okay? So the strength of my stimulus will be some sort of electrical charge, right? Let's say we start out with 50 millivolts, okay? I apply 50 millivolts to our kind of little neuron over here, and I want to see what happens to this guy's membrane potential. So I apply 50 millivolts, and guess what I see? I see a small bump upwards in this guy's membrane potential, okay? Whoop. But remember, I'm trying to test to see if I can generate an action potential, okay? So this is my threshold potential, right? That negative 55 millivolts that I have to hit, right? Let's say that, again, these are just random numbers. I apply 50 millivolts into this cell, and we get something like this. Let's say I up the voltage. I up the voltage a little bit, and what happens? Let's go to 100, okay? At 100, I get a slightly higher one, slightly higher graded potential response. Let's say I go to 150, I get an even slightly higher response. Let's say I go to 200. Okay, at 200, what do you notice? I hit the threshold and an action potential occurs. Great. Great. So that's one action potential. But notice something else. Let's say I up my strength of my stimulus to 400. What do you notice? What do you notice? I hit the exact same magnitude. I went up to the exact same place. Even though I upped my strength, nothing happened. Let's say I go up to 700 millivolts. What happens now? Same thing. I don't actually go any higher. What does this tell you? What does this whole part of the experiment tell you? This tells me that the amplitude, okay, the from here to here portion of the action potential stays the same after I hit the threshold stimulus. Regardless of how far I increase my stimulus, just as long as it's hitting that threshold stimulus, I will launch an action potential, okay? And it won't matter if I increase it all the way up to like 700, right? I'm just wasting my time. I always generate the exact same response, okay? So whenever I hit my threshold, I call this the threshold stimulus because this guy got me the threshold. And you notice that it doesn't matter that if I keep increasing the threshold stimulus, right? It doesn't matter. Like I will generate the exact same action potential every single time. These guys that did not get me the threshold, they're called sub-threshold stimuli. Okay. All right. And the sub-threshold stimuli, what do you notice? They're producing sub-threshold potentials. Threshold potentials. So the whole point of this experiment was to just prove to us or to ourselves that action potentials are indeed very consistent, okay, in their magnitude. And the way we proved that was by grabbing a neuron and continuously stimulating it with stronger and stronger stimuli, all right? And we figured out that after a certain point, which is our threshold stimulus, doesn't matter how much I increase the strength of the stimulus anymore, I will consistently get the exact same response. All right? So that's how we figured out that action potentials are very consistent. Okay? All righty. Next thing. Let's talk about this. Let's talk about this graph here. But before we do that, let's briefly say something. How is an action potential initiated? Okay? So the action potential is initiated, if you remember from our previous video, right? It starts with the summation of EPSPs, which are just a kind of graded potentials, in the cell body or dendrite, okay, of our post-synaptic neuron. And this will get us to threshold potential, right, which was negative 55 millivolts. Now, again, keep in mind, this might not always happen, but since we're talking about how is the action potential initiated, we have to say the summation of the EPSPs, right, which we're assuming had enough strength to get us to the threshold potential, will generate an action potential, okay? Give us a threshold potential, and then we lead to an action potential. So that's how an action potential is initiated. And we can kind of depict this using our graphical representation. So let's say right here is my resting memory potential, so negative 70 millivolts. I have EPSP, EPSP, EPSP. They are summing on top of one another, whether with temporal or spatial temporal or just spatial, we don't know. They are summing. And then I have some IPSPs in here as well, bringing my memory potential down, because we know that one neuron always has multiple kind of inputs on it. And then we have some other EPSPs that finally bring us to threshold, right? We said that this entire thing was called the grand post-synaptic potential, which was the combination of EPSPs and IPSPs that were acted on a postsynaptic neuron. We hit threshold. At threshold potential, we always get an action potential. It is an all or none phenomenon, okay? It's like pulling a trigger of a gun. You can't suck back in the bullet. It will just leave and, do its thing, okay? So this is the action potential graph, okay? It is about, let's kind of drag this down here. It's about 1 millisecond in duration, right? 1 millisecond in duration. These parts all the way here, these are the graded potentials that had to happen to get us to threshold, right? Or prevent us from getting to threshold, but in this case, get us to threshold via our ligand-gated channels. Okay. And the graded potential has many different the action potential has many different phases. So this first phase over here, what do you notice? We're going from a resting membrane potential, right, all the way up to a very, very high membrane potential. In fact, we're going from negative 70 millivolts all the way up to positive 30. And, we know that our threshold is negative 55. Remember how I said the membrane reversing membrane potential with an action potential? Well, that's what's happening here, guys. That's exactly what's happening here. I'm reversing the membrane potential from negative 70 all the way to positive 30. That's insane. It's really crazy. So what do you think is happening here that's getting us so close to positive 30? What do you think is happening? We are increasing the membrane potential. What was that called? What was that called when we're increasing the membrane potential? We said that that's called depolarization. So this right here is the depolarizing phase of the membrane potential. Okay? Afterwards, you can see that it's going back down all the way towards the resting membrane potential again. What is that called? That's called repolarization. And then you see the slight dip downwards from the resting membrane potential to a more negative area, about negative 80 millivolts. What is that called? We said that that was going to be called hyperpolarization. Or sometimes on this graph, they call it the after hyperpolarization event. The point is, we have a depolarization, repolarization, and an after hyperpolarization event. Okay? These are the phases of the action potential graph. And every single part of this has a different ion associated with it. Okay? So with this portion of the graph, it is just sodium movement, right? Because remember, sodium is one of the only ions we know that likes to come into the cell down its concentration gradient and make the cell positive. So this depolarization must be being caused by sodium. Okay, so sodium influx. Influx means inward movement, okay, into the cell. This red portion, repolarization. What ion do we know that is positive and likes to leave the cell, which will then make the cell more negative? Well, that has to be potassium. So this is potassium efflux. Efflux means exiting. E for exit, I for inward. Okay? Alrighty. Amazing. It's amazing. Now, remember, this entire thing is the action potential. Okay? And if you were to kind of draw a line from here all the way to here, right, negative 70 all the way to positive 30, you would realize that this is the 100 millivolt amplitude that we talked about. Okay? So we said 100 millivolt, very large amplitude. We talked about that. We talked about the fact that it reverses the membrane potential. Negative 70 to positive 30 millivolts. Okay? Great. We also said that it's very brief and rapid. 1 millisecond. Okay? Great. Now we're talking about all the ions that are responsible for it. The sodium influx causes depolarization. The repolarization phase is caused by potassium efflux. In fact, you'll see that the repolarization comes all the way back to the resting membrane potential, because that's its definition, going from positive to RMP. But then you'll notice the hyperpolarization is also in red, which means it's also caused by potassium efflux. We'll talk about what that is caused by as well. Okay? How is it that the potassium is actually leaving? Okay, great. So we'll finish on this graph for now, and let's come back here. Let's discuss these voltage-gated channels and how are they actually related to this graph, because they are extremely related to this graph. First things first, the sodium voltage-gated channel. What is its kind of characteristics? Well, the gates of this channel, they all start moving, okay? Gates all move at negative 55 millivolts, which is our threshold potential. So at threshold potential, all of the gates of the sodium voltage-gated channel are going to start to move, okay? But in which direction? Well, at threshold, there is a very rapid opening of the activation gate, okay? at threshold. At threshold. So when I hit negative 55, this activation gate snaps open. It snaps open. What about the inactivation gate? Well, there's actually a slow closing of inactivation gate. Okay? And in fact, it takes 0.5 milliseconds for it to close from the start of threshold potential. Okay, so as soon as we hit threshold potential, the timer starts, and after 0.5 milliseconds, the inactivation gate finally closes. All right? What do they look like at resting membrane potential? Well, at resting membrane potential, this is what they look like. The activation gate is closed, and the inactivation gate is open. Okay? Beautiful. Which phase of the action potential is this responsible for? Well, we know that sodium influx is responsible for the depolarization because it's making the membrane more positive. And this is, as a matter of fact, the sodium voltage-gated channel. So, put 2 and 2 together. The sodium that is influxing, coming into the cell, must be coming in through the sodium voltage-gated channel. So this is responsible for depolarization part of the action potential. Okay? I cannot stress the point enough, guys, that this channel does not depend on ATP. Okay? It is a passive channel. Voltage-gated just means that these gates that it has, they open at a specific voltage. That specific voltage is negative 55 threshold potential. Beautiful. Now, let's move on to the characteristics of the potassium voltage-gated channel. When does its gate open? Well, the sodium gates all start moving at negative 55. But we know that specifically the activation gate snaps open fast and the inactivation gate starts closing really slowly. All of this happens at threshold potential. With the potassium voltage-gated channel, the activation gate opens very slowly at threshold potential. Okay? Negative 55. And in fact, not only is it a slow opener, okay, not only isn't it a slow opener, it's also very slow at closing. Okay? And it is its configuration at resting membrane potential is like this. It's exactly like this, where the activation gate is closed. Okay? Closed. Now, which phase of the action potential is it responsible for? Well, we said that the potassium efflux is what's going to bring our membrane potential from positive 30 all the way back down to resting membrane and even to negative 80. Okay? So it's going to be responsible for repolarization and the hyperpolarization phase. So repolarization and the hyperpolarization. Okay, great. Now that we know all of this, okay, let's kind of talk a little bit more about this graph. This graph has some other parts to it, okay? It's got some other parts to it, where this portion of the graph, from here all the way to here, is called the absolute refractory period. And this portion of the graph is called the relative refractory period. We'll talk a little bit more about this later, but for now, I really just want you to understand something very simple, which says the absolute refractory, or the absolute refractory period, literally just means that we absolutely cannot launch an action potential from this time period to this time period, which means that during the action potential, when the action potential is happening, we can't just launch another action potential. This is not how it works. Okay, so I can't for some reason when I'm at the peak point here just launch another action potential. That's just not how it works. It doesn't work like that, and we'll talk about why. And the relative refractory period, well, this is relative. That means that I can actually launch an action potential here, but it's relative. It depends. What does it depend on? It depends on the strength of my stimulus. So remember how these were all my stimuli that got me to the threshold potential? Well, this was going from negative 70 to negative 55. But I'm not going to lie to you, it's a little bit harder to go from negative 80 all the way to negative 55. It takes a bit more juice. It takes a bit more strength. Okay, so I can launch an action potential from this time period to this time period, but it really depends on whether or not the strength of my stimuli can take me there or not. Okay, if it can, then great, I can launch another action potential here. If it can't, then I'm not launching an action potential. Okay, great. So that's all for the absolute and relative refractory period for now. Now, I want to do something here, and I want to go ahead and take a look at this graph over here, where we're going to correlate these voltage-gated channels, and we're going to look at which point of this graph they are either open or closed. Okay, so basically we're going to look at their configuration at different portions of this graph. Okay, and by the way, now that we've mentioned these voltage-gated channels and that they open at threshold potential, we can kind of change our definition of how the action potential is initiated and make it a little bit more accurate. We said that it is the summation of EPSPs in the cell body or dendrite of our postsynaptic neuron. This will get us to threshold potential, and then action potential occurs. We can modify this even further and say that this will get us to threshold potential, which opens / moves the gates of our voltage gated channels. Okay? Responsible for the action potential. Okay? Great. So you can see how we can expand on this definition much, much more now that we know more about the process. Okay? And I urge you to do the same thing as we go through this video. Okay? Try and expand this definition for yourself based on what we keep talking about. Okay? Alrighty. So now, remember, we have our depolarization phase, repolarization, and the hyperpolarization phase. Okay? And this is, of course, just our triggering event or our graded potentials. We know that the peak of depolarization is positive 30. We know that our threshold potential is negative 55. And we know that our resting membrane is negative 70. And we know that at the end of hyperpolarization, we come to negative 80. Okay, let's go through it. So, number one, what does our kind of channels look like? We know that this is the sodium voltage gated channel, and we know that this is the potassium voltage gated channel. We know that at resting membrane potential, what is the configuration? Well, the inactivation gate is closed while the inactivation gate is open while the activation gate is closed. And for potassium, we know that the potassium voltage gated activation gate is closed. Okay, this is all kind of what we said in the last slide. Now, after we hit threshold potential, what happens? We know that at threshold, all gates move, all of them. Okay, so you'll see that the activation gate immediately snaps open for the sodium voltage gated channel. For the potassium voltage gated channel, it slowly starts to open. Okay? Now, how can I depict that here? I like to depict it with this arrow here that shows slowly opening in this direction. Okay? Now, this guy, what about this guy? Well, it's slowly closing. But it's closing in that direction. Okay? Now remember, how long did I say it takes for it to close, guys? It takes 0.5 milliseconds. Remember this number. This is really important. But while this is closing, this activation gate is open and the whole channel is open. So that means that in this scenario, sodium can easily come in. And this guy we know again is slow opener. Okay, now look at this. Now, this sodium will continue to keep rushing in from these voltage-gated channels until we hit positive 30. And at positive 30, for some reason, it stops coming in. Sodium stops coming in. You might be thinking, hey, Kian, don't you think that all of the sodium should keep coming in until we hit positive 65 millivolts, which is the equilibrium potential for sodium? And you'd be correct if this gate remained open, if this channel remained open. But remember, the entirety of the action potential lasts 1 millisecond. The inactivation gate closes after 0.5 milliseconds. So 0.5 milliseconds is exactly the halfway point of this action potential graph. So at this halfway point, what do you notice? You can notice that at this halfway point, this inactivation gate closes. Let's draw it a little bit better. This inactivation gate is closed at this halfway point, guys. Okay, and that's why sodium can't come in anymore. Because watch, here's that sodium, right? It tries to come in, but it can't. And you also notice that the activation gate is still open, but the inactivation gate is the one that's closed now. Okay? Now, at this point in time, the potassium voltage gated channel also coincidentally happens to have fully opened. Because remember, it was starting to open here, but now it's fully open here. And now what do you notice? Now you can see that the potassium will just leave down its concentration gradient outside of the cell, just like we've been talking about. And it will keep going until we repolarize all the way down to the resting membrane potential. One note I do want to add on here, though, is that potassium is not only moving out via its concentration gradient. You know how usually at the resting membrane potential, potassium is coming in against its electrical gradient, but with its concentration gradient. In this scenario, though, because the membrane potential has fully reversed from negative to positive, potassium is being pushed out at this peak point due to a high electrical gradient and a high chemical gradient. Okay? So it's moving with its electrochemical gradient here. All right. Now, potassium will keep moving down until it's changing the membrane potential fully back to resting membrane. And then it will change it some more and bring us to this hyperpolarization phase. Why do we get to this hyperpolarization phase? Well, let me tell you, at this point, right, the entire time this guy has been closing. This activation gate of the potassium voltage gated channel, it's been closing. But what do you remember? What do you remember? I said that this guy is a slow opener and a slow closer. So he took a long time to open, but now he's taking even longer to close. If he's going to take super long to close, guys, what's going to happen? A little bit more potassium that we would like would actually keep leaking out. And so we will go from resting membrane potential, we will pass it, and we will reach negative 80 because this gate is so lazy that it took so long to close. Okay? Slow closer. And that's what causes the hyperpolarization effect. The fact that this channel, this activation gate for the potassium voltage gated channel, closes so slowly that we actually let out a little bit more potassium than we would have wanted to. Okay? Now, what is the configuration of the sodium voltage gated channel here? Well, guess what? As soon as we hit this resting membrane potential, the configuration of the sodium voltage gated channel returns back to what it was before, where the inactivation gate is open and the activation gate is closed. Okay? So from here all the way to this point right here, it stays exactly like this, but then as soon as I hit this point, it switches and the inactivation gate opens up and the activation gate closes. Okay? Coincidentally, this right here, the fact that this configuration changed is why in this scenario to this scenario, we can actually launch another action potential. It's all about the configuration of the sodium voltage gated channel. Okay? It's because this guy changed and the activation gate can now be opened again. Whereas over here, the activation gate was already open. And no matter how much stimulus I give here, the inactivation gate will never ever open again unless we go back to the resting membrane potential. Okay? Again, we'll talk about this a bit more towards the end of the video, but this is actually the kind of cellular level reason for the absolute and relative refractory period. Okay? Now, one last thing I really want you to understand here is that the membrane permeabilities change all throughout this action potential. So remember that at resting membrane potential, right? In my membrane potential video, I said that the membrane is 50 to 75 times more permeable to potassium than it is to sodium. Okay? But all of that changes as soon as the action potential starts. And why does that change? Because of the voltage-gated channels. From steps 2 all the way to step 3, the membrane permeability favors sodium. Why does it favor sodium? Because from steps 2 to 3, all of the sodium voltage-gated channels on the membrane are open, but the potassium voltage-gated channels are closed and are actually very, very, very slowly opening. Okay? So the membrane permeability from steps 2 to 3 looks something like this. The membrane is 600 times more permeable to sodium than it is to potassium. Okay? Membrane permeability from step 3 all the way to step 4 looks a bit different as well. In this scenario, all of the sodium voltage-gated channels are actually closed, and the potassium voltage-gated channels are now fully open and are going to slowly close. Okay? Which means the membrane is now way more permeable to potassium than it was to sodium. In fact, it's 300 times more permeable. Okay? So it's really important to understand that kind of permeability scheme over there. All right? Great, great. Now that we understand how the action potential graph looks like, and that an action potential is really just kind of caused by these voltage-gated channels, right? And we know what kind of triggers an action potential, right? It starts at threshold, right? Which, what got us to threshold? It was the graded potentials. We can now kind of start looking and zooming out and looking at an overview of how is the action potential actually conducted throughout the length of a neuron, okay? So remember, here was our neuron, right? We kind of zoomed in and looked at an action potential on just this one section. But now we have to look at how is that action potential going to get conducted throughout the rest of that neuron. And that's exactly what we're going to do right over here. There are two ways to kind of conduct an action potential. Okay? So once an action potential has started, there are two ways to conduct that action potential. And really, those two ways depend on the kind of neuron you're dealing with. Okay? And we'll talk about that in a bit. But for a second, just bear with me. Here's the overview of conduction. Okay? The charge, the action potential, travels in one direction and one direction only. We say that it is one-way propagation. So graded potentials will bring us to threshold potential, which is negative 55. And right here in this portion of the neuron called the axon hillock, the action potential starts, okay? And that action potential will keep spreading throughout the membrane, and you'll notice a depolarizing wave that will kind of look like this. So these guys in blue are the sodium voltage gated channels that are responsible for the depolarizing wave. And then following right behind it is the repolarizing wave caused by the potassium voltage gated channels. Okay? This is an overview of the conduction pathway. We'll zoom in on this and go into greater detail in a second. But briefly, why is it that the action potential starts at the axon hillock? Well, it just has to do with the anatomy of the neuron, right? We receive our input, our graded potentials, right, on our dendrites or on the cell body. So when those guys are enough to hit threshold, guess what? Those positive charges that come in will travel along the membrane and they'll reach this area. You know what this area is? This is the axon hillock. Why does the action potential start here? This axon hillock has the greatest density of voltage-gated channels, both sodium and potassium. So if it has the highest density of these voltage-gated channels, then an insanely large amount of change in membrane potential will occur here, and that will just kind of propagate and spread throughout the rest of the neuron, specifically the axon, and go all the way down towards the synaptic terminals. Okay. Now let's look at how we actually conduct this action potential. Okay? There are two ways of conducting an action potential. The first way is actually called saltatory conduction. Okay? The other one is called contiguous conduction. Okay? Saltatory conduction, a couple of things about it, is that it is very fast. Okay? It is extremely fast conduction speed. All right? And that is because the neuron, all right, the actual neuron has this thing called a myelin sheath. okay, which is really just 80% lipid and about 20% protein, okay, and 20% protein. But that's what a myelin sheath is. It is literally just 80% lipid and 20% protein. And because it is so much lipid, it acts like an insulator. And if you guys know anything about insulators, they like to increase conduction velocity, conduction speed. Okay, so fast, speed, conduction. That means the action potential goes really fast, really, really fast. Okay, so always associate the presence of myelin on a neuron with fast conduction. And the type of conduction is called saltatory. Okay, it just describes how fast it is because of the presence of the myelin. Now, another characteristic of a neuron that has myelin is that they also have these things called the nodes of Ranvier, which we'll look at. Okay, but the nodes of Ranvier, they're just really 1 millimeter spaces between adjacent myelin sheaths. Right? So let's take a look at a neuron with myelin. Here is a myelinated neuron, and here's an unmyelinated neuron. And what you'll notice is that the myelin is just surrounding the actual axon, okay? So this one, there is no myelin, right? Here is the myelin, here's another myelin, myelin, myelin. Where are the nodes of Ranvier? If you can see, there is a tiny space in between each of these myelins. That space in between them, where there is no myelin actually covering the axon, right? And the axon is kind of naked like this, that space is called the nodes of Ranvier. All right, and that node of Ranvier is about 1 millimeter big. All right, that's the length of it. It is the space between the adjacent myelin sheaths. And if somebody were to kind of look at this conduction, this saltatory conduction, what they would notice is that the action potential seems to be kind of jumping from one node of Ranvier to another node of Ranvier. And we'll talk about why this kind of phenomena seems to be occurring. All right? And finally, the other thing is this guy right here, a myelinated neuron, has a very, very low concentration of voltage-gated channels on it. What do I mean by this? Why? Well, if I have this giant myelin, this fatty kind of lipid myelin just surrounding this axon, all right, the plasma membrane of that axon, do you actually think that I'll be able to plug in any voltage-gated channels where these guys are sitting? No, these guys are occupying all that space, okay? So because of the presence of this myelin, the only place that has voltage-gated channels is the nodes of Ranvier, right? And because of this, compared to an unmyelinated neuron, which has no myelin, the myelinated neuron has a very low concentration of voltage-gated channels. So low concentration of voltage-gated channels. Okay. The same thing can be said about unmyelinated, okay? Unmyelinated neurons have something called contiguous conduction. You want to know how I remember this? I like to remember the contiguous is very, very similar to the word continuous. What does that mean? Remember how I told you in saltatory conduction, it seems to be that the kind of the action potential is jumping from one node of Ranvier to another? Well, guess what? In an unmyelinated neuron, there is no node of Ranvier. So what happens? There's a very smooth, continuous flow of the action potential. Okay? That's how I like to remember that continuous, right? That contiguous has a continuous flow of the action potential, and it must mean that it is in an unmyelinated neuron. Okay? Because there's no myelin to kind of get in the way and stop the flow of current from being continuous. Okay? That's how I remember it. Now, if there is no myelin, right, the conduction speed is much slow. Okay, so slow conduction. Okay, and that's because there is no myelin. No myelin. But there is a continuous flow of ions, okay, along the axon. Um, and once again, because there is no myelin, there is a high concentration of voltage-gated channels. Okay, great. Again, it has to just do with the fact that there's no myelin. All right, so we keep talking about myelin. Let's give an example of a myelinated neuron. So this guy right here is a great example of a myelinated neuron. And you can really kind of just think about this. A motor neuron, right, which we'll talk about in the next video, a motor neuron is a great example of a myelinated neuron, right? Why would I say this? Because a motor neuron is responsible for contracting your skeletal muscles, right? And if a myelinated neuron has a very fast conduction speed compared to an unmyelinated neuron, then it makes sense. It makes sense why we would want to use this to kind of, innervate our skeletal muscles. Because imagine you think about snapping your fingers, right? Can you imagine how weird that would be if you thought about snapping your fingers and it happened 5 seconds later? The only reason that would happen is if you have a very slow conduction velocity. But myelin solves that problem. Myelin is very, very fast, okay? Not very, very fast. It speeds up the conduction velocity of the neuron. So when you think about snapping your fingers, it happens almost instantly, okay? In fact, the presence of myelin can increase conduction velocity by 50 times almost, okay? And of course, there's levels of myelination. Some neurons are more myelinated than others, okay? Now, where does myelin come from? Where does myelin come from? Well, myelin can come from two different places, and it depends where you're looking, right? If you're looking in the central nervous system, which is just your brain and spinal cord, right? Those neurons that are myelinated in that region of your body will have been myelinated by a cell called an oligodendrocyte, okay? Oligo means many. Dendrocyte means that it has these kind of arm-like extensions. So, of these two, which one looks like an oligodendrocyte? I'd say this one. This one is an oligodendrocyte, okay? And oligodendrocytes are so interesting. They are the cells that actually produce myelin in the central nervous system, okay? And they can myelinate up to 20 to 30 axons at a time, simultaneously. So look, he's doing it right now. One neuron he's myelinating, another neuron he's myelinating, and another neuron, right? These are all separate axons. This is another axon, okay, and another axon. What about the peripheral nervous system, which includes your autonomic nervous system, your somatic nervous system, right? So somatic nervous system would include your motor neuron, right? So let's talk about how this motor neuron got myelinated. It was myelinated by a cell called a Schwann cell. This Schwann cell is very interesting, okay? So here it is, Schwann cell. The Schwann cell itself will wrap around the axon of a single neuron, right, multiple times. And you can see here's its nucleus, the Schwann cell's nucleus. And it will finally produce myelin, okay? And unlike the oligodendrocytes, which can be myelinated multiple things at once, the Schwann cell can only myelinate one cell, okay? One neuron, I should say. So myelin originates in the central nervous system from these cells called oligodendrocytes, the PNS, Schwann cells. Okay? Great. And of course, here's a better view of your node of Ranvier, right? This is a node of Ranvier. This is another node of Ranvier. Okay? And of course, I didn't show it here, but there would also be another node of Ranvier. Okay? Great. Great. So this is the origin of myelin. This is where myelin is actually coming from. All right? Great, great, great. Now, let's kind of zoom in on this image that we were talking about and look at conduction. But let's look at conduction in two different ways. Let's look at it in a non-myelinated neuron, or an unmyelinated, and a myelinated neuron, okay? So let's write this down for ourselves. This is unmyelinated. This one is myelinated, okay? Alrighty, let's take a look. So in a unmyelinated neuron, right, what has to happen first? we start at the axon hillock, right? And let's just say this is the axon hillock and this is the rest of the axon, okay? This is the rest of the axon, okay? Well, what has to happen to the axon hillock? Well, the axon hillock has to reach threshold potential, correct? Because that's how an action potential starts. So the threshold potential must be reached, which is at negative 55 millivolts. Okay, great. And that is going to do what? That is going to open up these channels, or rather move all of the gates of these channels. So this was our sodium voltage gated channel. This is our potassium voltage gated channel. And if you remember, at threshold, what would happen? The activation gate will snap open, okay? And the inactivation gate would slowly start to close. Whereas with this guy, you would notice that the its activation gate would slowly start to open, okay? And in that process, what was happening? A lot of sodium was moving down its concentration gradient into the cell, making the cell super duper positive. Okay? Now, this area of the cell, only this area of the cell, would go from negative 55, which is where it was at now, right? It would go from negative 55 millivolts all the way to positive 30 millivolts, okay? As this guy will continue to bring sodium in. Then what happens? Then at positive 30, we know that this gate finally closes. The sodium influx stops. These positive charges are currently still here, by the way. And this gate fully opens now. If this gate fully opens, what's going to happen? Potassium will now start going out, okay? And making the inside of the cell negative, repolarizing, hyperpolarizing, okay? Which means what would happen to the voltage now? Well, you will then go from positive 30 millivolts all the way to negative 80 millivolts, okay? So that's what's happening here. And this entire portion right here at the axon hillock underwent one action potential, okay? It underwent one action potential. But guess what? I said the action potential regenerates itself along the membrane, right? How does it do that? Check it. Those sodium ions that came in, those positive charges, they don't just sit here, man. Once this area is repolarizing and hyperpolarizing, those guys are moving on. Where are they going? They're going to come here. And they're going to bring this area of the cell, which was at negative 70 millivolts, by the way, they're going to bring it to threshold. Okay? And once they bring this area to threshold, what's going to happen? The gates of these channels will open. Okay? I'm not going to redraw it because you guys should know that now. But the gates of the sodium voltage gated channel will start moving. Specifically, the activation gate will blast open. The inactivation gate will start to close. And sodium will come into the cell, depolarizing this area. Okay? And bringing the membrane from negative 55 millivolts to positive 30 millivolts. Okay? And then what's going to happen? Then the inactivation gate will finally close. The activation gate will still be open, okay? But at that same moment in time, the potassium voltage gated activation gate will finally open. And what would happen? Potassium will then start going down its concentration and electrochemical gradient down until it makes the cell super, super negative, bringing us from positive 30 millivolts all the way down to negative 80 millivolts, okay? So now this portion of the membrane underwent an action potential, okay? Guess what? This positive charges that came in, they're not going to sit here either. While this area is repolarizing and hyperpolarizing, they will have been moving on, okay? And activating this guy, all right? Now, you might be asking me, hey, Keon, these positive charges here, I understand that they came and kind of brought this area to threshold and they opened up the gates of the sodium voltage gated channel. But how does this guy open up? It's the same thing, man. These positive charges, they don't just kind of bring the sodium voltage gated channel to threshold. They bring this entire area to threshold, all right? Meaning that this positive charges will also activate these guys, okay? And so will this. Look, this guy will come here and activate both of these. And then same thing, man. Sodium will come in, make the inside super, super positive, okay, until we're at positive 30 millivolts. At positive 30, the gate, inactivation gate closes. The potassium activation gate opens up, and then it moves out, okay? Making the inside negative until we're what? From positive 30 all the way to negative 80 millivolts. Again, those positive charges, they wouldn't have just stayed there, right? Once they came in, they would have started to move along the membrane, okay? Great, and they would have done the same thing. I'm not going to finish the rest of this because it's the exact same scenario. It's literally the exact same scenario, all right? Okay, now, here is the big question, okay, that I think everybody should and needs to understand. Once this area starts in an action potential, right? Let me draw the graph for you. This is how it would look like. So boom, boom, okay? And I'm going to copy this over for the rest of them so you understand. Okay, so when I open up the gate of this guy, right? When I reach threshold and the activation gate blasts open and sodium starts coming in all the way towards the peak of depolarization, right? You know where this area currently is at? This area is currently right here at resting membrane potential, okay? But let's move this area now. Let's move this area to here. Let's say this guy's starting to repolarize. Where would this area be now? This area would now be here. He will have already gone through, the threshold potential and started depolarizing already. But this guy's currently at resting membrane, and this guy's at resting membrane. Okay? Now, when this area finishes, its hyperpolarization and is here, this area will now be oh my God, I'm so sorry. This area will now be repolarizing, and this area will now be in its action potential phase. Okay? I'm just going to kind of get rid of that. There we go. The whole point I'm trying to kind of depict to you is that when we describe this singular action potential graph, right? That's all it is, a singular action potential occurring at a singular region. But multiple of these have to occur within the entire axon, okay? Again, we imagine this was the axon hillock and this was the rest of the axon. And this will continue carrying forward all the way down the entirety of the axon and spread throughout those synaptic terminals. We'll talk about that in a second. But I really need you to understand that, okay? Great. Now, before we move on to a myelinated conduction, which is extremely similar, by the way, let's talk about the one-way propagation of the action potential. Why is it one way? What contributes to it being one way? Well, the one-way propagation, the main reason is the refractory period, the absolute refractory period. And I want you to recall that the conformation of the sodium voltage-gated channel at the peak of depolarization, right here, right? It has open activation gate, closed inactivation gate, okay? This configuration prevents the re-excitation of the previously depolarized area. So what do I mean by this? Let's take this scenario right here. This area is currently being depolarized, whereas this area is currently undergoing repolarization. What would the sodium voltage-gated channels look like then? So in this scenario, let's lower this. Sodium would be coming in, okay? And at first, what do you think happens, right? What do you think happens? These positive charges, they will kind of travel in this direction, right? They'll travel in both directions, actually. But first, they'll come in this direction and they'll try to re-excite this area. But you know what they'll figure out? They'll figure out, hey, the inactivation gate of this guy is closed. And no matter how hard I try, I cannot open this. Okay? This guy's repolarizing. And, in fact, another thing, this area would be much less negative than this area currently. Because this area right now is at resting membrane potential, whereas this area might be still repolarizing. So this area would still be a little bit positive as compared to this area. So the charges would also just get repelled this way, all right, and continue moving and propagating in one direction. That is why we have this one-way propagation. It's because of the configuration of the sodium voltage-gated channel at the peak of depolarization, okay? And this happens all throughout the axon, which is what I was kind of describing here. While this guy is depolarizing, this guy's repolarizing. So the positive charges that come here not only get repelled to go to a more negative direction, but they realize that they just can't open the inactivation gate here, okay? Great. So that's how the one-way propagation works. Now, let's finish up with the myelinated conduction, which was called what? Saltatory conduction. This right here was contiguous conduction. With saltatory conduction is more of the same, guys. It's literally more of the same. Threshold potential has to occur, negative 55, right? Millivolts. On the axon hillock, which we define as this portion, we will open up the gates, okay? Sodium will, of course, move in until it brings the cell from negative 55 all the way to positive 30, okay? At that point, potassium voltage-gated channel opens up, okay, fully, and the potassium will go all the way out until we are from positive 30 all the way to negative 80 millivolts, okay? The only difference here, guys, the only difference between this and this is that, remember, I told you that because there is myelin present on the membrane, I cannot fit a voltage-gated channel here. I just can't. I physically can't. It's impossible. So this guy acts like an insulator, okay, preventing any charge from even being here, okay? So this area currently has a lack of charge. Okay? And that's because it's completely insulated by this giant myelin that's like 80% lipid. So you know what happens to these positive charges that just came in? They don't have to stop on their way to open up this channel, right? Over here, they would have to keep stopping, opening channels, and then moving on. Over here, look at it. It just moves all the way here and opens up these guys now. Okay? Look at how far this guy traveled and how fast he traveled without having to kind of keep stopping, opening channels, moving on. Keep stopping, opening channels, and moving on. This is why myelin increases the conduction speed, okay? It literally just forces these charges to just keep moving on and traveling much faster towards the next location, okay? Great, beautiful. And again, the same thing will happen. These charges will bring this area towards threshold because it was currently at negative 70. It'll bring it to threshold. Then it will open the gates. Sodium will comes in. We'll go from negative 55 to positive 30. And then at that positive 30, the potassium voltage gated channel opens, right? Making the inside negative until we get from positive 30 to negative 80. Okay? And so on and so forth. This thing just keeps happening, okay? Boom. Boom, boom. Okay, great. So that's how myelinated conduction, aka saltatory, and unmyelinated, aka contiguous conduction, occurs. Last little note about conduction speed. So we know that myelin increases conduction speed, but there's another thing that also increases conduction speed, which is fiber diameter. So if I have a neuron whose axon has got a much higher diameter than the other one, this one will have a faster conduction velocity, okay? And that has to do with many reasons, but I think the basic reason is that if I have a very, very, very small kind of diameter, then the resistance to flow increases, all right? I always like to think of it like this. Where can water flow easily in? A very compressed small tube or a very large pipe? Well, it can flow much easily in a larger pipe. Thus, the speed would be much faster, okay? So if I increase the diameter of a fiber, that is directly proportional. By the way, this is a mathematical symbol that says directly proportional to increasing the velocity. If I decrease the diameter, then it will directly decrease my velocity, okay? Pretty easy, I think. Okay, now we are done with all of this kind of conduction and action potential. The last topic we're going to start to tackle here is this. What happens after that action potential has finally spread to our synaptic terminals. What happens then? I'll tell you what happens. So remember, we started with a presynaptic neuron right here that gave us some sort of threshold stimulus, right? EPSBs that would keep summating on top of one another, and it got us to threshold potential. And then on the axon hillock, we initiated the action potential, and the action potential just kind of spread all throughout the actual membrane, right? So it would keep going like this, and this. Now, this guy will keep traveling until it reaches these two locations, okay? And this location, this entire thing, is kind of a zoomed-in view of the synaptic terminal. Over here on the synaptic terminal, we will encounter specialized voltage channels again, but these guys are called calcium voltage-gated channels, okay? Now, you don't have to know which kind of voltage these guys opens at, or you don't have to know that it has a gate or whatever. None of that matters. Just know that this action potential, these positive charges from the action potential, will keep kind of moving until they open up these calcium voltage-gated channels, okay, at a very specific voltage, right? And once they do that, you know what's going to happen? Typically, calcium is very high outside of the cell and very low inside of the cell. So what will it do? It will move from outside to inside. Okay, same thing here. Now you might be thinking, hey, why would I even want this to happen? What good is calcium to me? Well, inside of the synaptic terminal, right, we have neurotransmitters stored, right? Remember that? We've talked about this many times. But where are these neurotransmitters actually stored? They're stored inside of these storage vesicles. If you remember, vesicles, in order to secrete their contents, they have to fuse with the cell membrane. Well, that required V snares and T snares, okay? Now, I could get into very complex detail here, but for the purpose of this video, I won't. The only thing I would want you to know is that calcium causes the binding of this V snare right here, okay, to this T snare, like this. And that causes the exocytosis, right, or the fusion of the vesicle with the membrane, and the exocytosis of the neurotransmitters outside. Okay? So what is this protein called? What is this V snare specifically called? This V snare is specifically called synaptotagmin. Okay? And honestly, you don't have to know how the calcium kind of facilitates the binding of synaptotagmin with this T snare. Okay? There are many proteins involved in this process, such as syntaxin, SNAP25, synaptobrevin. You don't have to know any of that. All I want you to know is that synaptotagmin, right, with the help of calcium that just came in from these voltage-gated calcium channels, right, will come and stimulate the fusion of the synaptotagmin and this T snare. And once that happens, fusion of the vesicle will occur and exocytosis of the neurotransmitters inside will happen. Where would these neurotransmitters go? Where are they getting released into? Well, if you remember, they get released into an area called the synaptic cleft. Because remember, every single neuron is going through a synapse. Okay? And in fact, this right here is a synapse between a presynaptic and a postsynaptic neuron. But as soon as this guy releases its neurotransmitters, and let's say there's another neuron here, he now becomes the new presynaptic neuron, and that other neuron becomes the postsynaptic neuron, okay? So we took a zoomed-in view of a synapse. What does it look like? Well, here is the, let's kind of increase our font. This right here is the presynaptic terminal, okay? Presynaptic terminal. These are the calcium channels, the calcium voltage-gated channels, right? Remember, calcium comes in and it stimulates the fusion of this vesicle with the membrane, which causes the release of neurotransmitters inside of this area. What did I say this area is called? This area is called the synaptic cleft, okay? Cleft. The synaptic cleft is a very specialized area. It's very small. It's around 10 to 20 nanometers, okay? Very, very small. 10 to 20 nanometers, okay? And inside of this area, these neurotransmitters that are released will interact with the post-synaptic dendrite or cell body membrane. And what I mean by interact with this, well, these neurotransmitters, right, they will go and find their very specific receptors, right? Remember, we talked about in the graded potential video, there were those ligand-gated channels. And we said there was two types. There was inotropic and metabotropic. This one, for example, is an example of an ionotropic, ligand-gated channel. So those neurotransmitters, they will go and bind to their very specific receptor. They will open it, and then they will allow either an EPSP or an IPSP to occur, depending on whether it's an inhibitory or excitatory synapse, right? So this whole thing is called a synapse. This area is called the synaptic cleft, okay? Where neurotransmitters are released and they will bind to receptors located on the postsynaptic dendrite or cell body membrane, okay? Pretty simple, I think, okay? Now, there are two types of synapses. This specific example is of a chemical synapse. Why is it of a chemical synapse? Because we're releasing a chemical stimulus called the neurotransmitter. Okay, so we have a chemical synapse. There is another one that I already have a video on, and this one is called an electrical synapse. Electrical synapses, the most common place you can find this is in the heart muscle, where there are gap junctions involved. So if you're interested, you can go watch my cardiovascular playlist. It's in there. But that's a other kind of synapse, okay? Gap junction. And we talked about what gap junctions are in the cell physiology video, okay? This chemical, again, involves neurotransmitters, which is what we are interested in, okay? But here's the thing. Do these neurotransmitters always just kind of sit here inside of the cleft? Can't be, right? No, they get removed. And there are three ways of removing these neurotransmitters. And by the way, these ways, they are not mutually exclusive, meaning that some of them may occur within one synapse or only one might occur within a given synapse, okay? We don't know. It just depends on the example. One of the first ways, I'm going to do one, I'm going to do two, and then I'm going to do three. One of the first ways is, and let's do it like this. One of the first ways is by enzymatic degradation, okay? So you will take this neurotransmitter who is red and active, and then we will turn it inactive, right, into this kind of green inactive thing with the help of an enzyme. This enzyme might be secreted from here, or it might be secreted from here, right? But it will reside inside of this cleft, right, the synaptic cleft, and it will degrade any neurotransmitters that haven't actually kind of, done their thing. An example of this is an enzyme that loves to degrade acetylcholine. It's called acetylcholine esterase, right? So acetylcholine esterase. That's the enzyme that resides in a lot of synaptic clefts that have acetylcholine involved. Other times, we will have active reuptake, okay? Active reuptake of the actual neurotransmitter, right? Whether it uses ATP or it uses some form of, secondary active transport, okay? We will actively reuptake the neurotransmitter back into the presynaptic terminal, okay? Where over here, it will be broken down into its constituent parts and then recycled and then, reused again. Oftentimes, for example, acetylcholine can be taken back up, okay, broken down into an acyl and a choline, okay, go into the mitochondria and then become acetylcholine again, and then become packaged into another vesicle, ready for kind of secretion again, okay? And the final thing is that sometimes these neurotransmitters can just diffuse away from the synapse. They can literally just diffuse away from the synapse. And if we're talking about the central nervous system, there are these specialized cells called astrocytes, okay, which just help support the function of neurons. These astrocytes will pick up these stray neurotransmitters, okay, and then, degrade them, recycle them, and then kind of reuse them, okay? I will give you a clinical significance here, okay? If you guys have heard of selective serotonin reuptake inhibitors, right? So selective serotonin reuptake inhibitors, they're used to treat depression, quote unquote, okay? And the way they're used, or their mechanism of action is, the neurotransmitter is serotonin, right? Which is responsible for this kind of like happy moods, right? The idea is, if I prevent the reuptake of serotonin from the synaptic cleft, it remains bound to its receptor longer, and it can signal that kind of happy feeling for a longer period of time, okay? And so what they do is they give SSRIs. These drugs will do exactly this. They will block step 3, this third way of reuptake. They will block the serotonin reuptake channels, okay? That's what they call selective serotonin reuptake inhibitors. Okay, so that's the clinical significance here. Alrighty. The last thing that I really do want to talk about with regards to the synapse is the concept of postsynaptic integration, which we really did touch on a little bit in the graded potential video, but the notion of postsynaptic integration is really important, okay? Because let's say I have one stimulatory synapse happening right here, meaning if this guy launches an action potential, it results in an EPSP here. Let's say I have an inhibitory synapse also occurring on the same postsynaptic neuron. If he launches, he will create an IPSP. But then I have these weird scenarios where I have a stimulatory synapse occurring, but then an inhibitory synapse is already in place with this stimulatory neuron. You know what this is called? This is called presynaptic inhibition, okay? Because this dude is going to try and launch an EPSP, okay? But before he even does that, right, his action potential has to get down, right, to its synaptic terminals. This guy is kind of acting like a hater, okay? He is just launching an inhibitory synapse right over here to try and kind of neutralize anything that this guy wants to do. That's called presynaptic inhibition, all right? This one right here is called presynaptic facilitation, okay? Because it's going to facilitate a bigger kind of EPSP, okay? And you see that here. It's going to try and enhance the stimulus. Let's go through an example. So we have neuron A, neuron B, neuron C, neuron D, neuron E, and neuron F. E and F are currently within a presynaptic facilitation. C and D are currently within presynaptic inhibition. We have our resting membrane potential here, we have our threshold potential here, and we have our peak of depolarization here. Our goal is to kind of figure out how this postsynaptic neuron is receiving all of these stimuli, okay? That's the notion of postsynaptic integration. We have to integrate all of our kind of responses to figure out what our final response will be. So if I just launch A, what will happen? I will get a rise in the membrane potential, a depolarization, and then a subsequent repolarization. If I fire A and B together, assuming that they have the same magnitude, they will cancel each other out, okay? What if I fire C and D together? Same response. Because they're going through presynaptic inhibition, they will cancel each other out and we will remain electrically neutral. Or that's at least the reading that we will get on the postsynaptic membrane. But what if I fire A twice? What if I do temporal summation? Okay, this is temporal summation, right? If I fire A twice, that's temporal summation. Well, I would get a bump and another bump. What if I do E and A together? E and A, so excluding F. That's spatial summation, okay? If each of them fires once, boom. What if I fire E and F? Okay, so E fires and then F fires at the same time. Then you will get a very smooth and continuous rise. Okay, so that presynaptic facilitation is very powerful. And that would get me to threshold, and then I would go all the way up and then repolarize again and then come back down. Okay, to resting membrane potential. But then what if I launch B? Well, that would give me a hyperpolarization. Okay, now again, this is not all of the combination or possibilities by any means, but I just really want to introduce the concept of postsynaptic integration to you, as I did back in the greater potential video, because it's really important to understand that a neuron receives many, many inputs, because that's exactly how our nervous system is. That's how our brain works, right? That's how all of our nervous system works. All of our neurons are constantly receiving many, many inputs. In fact, the dendrites in the cell body of a given neuron can receive as many as 100,000 synaptic inputs. That's very impressive, okay? So keep that in mind when, just learning this kind of concept. Guys, we went through a lot of concepts here, okay? We talked about the action potential in depth. We talked about what happens when the action potential finally reaches the synaptic terminal. We talked about neurotransmitter release and then removal out of the synaptic cleft. And we talked about the notion of postsynaptic integration, when the neurotransmitters have all been kind of released and are acting on the postsynaptic membrane, okay? I hope this all made sense. If it really did, if it truly helped you, hit that like, hit that subscribe button, maybe even leave a comment. It really does help with the channel. And I'll see you in the next video when we finally start discussing our nervous system organization and function.